Hello, everyone. On behalf of the editorial office of BMEH, welcome to today's webinar, and we are delighted to have you all here. To ensure a smooth session, we kindly ask everyone to keep your microphone muted during the presentation. Today's session will begin with a presentation from Professor Sabine. It's now my pleasure to hand over to our host, Professor Xu. He is from Suzhou University. Dr. Xu, the floor is yours.
All right, thank you, Jenny. Yeah, I'm from Suzhou University. I'm Congcong Xu. Yeah, I'm glad to introduce today's speaker, Professor Sabine, and her presentation title is Graphene-Based Field Effect Transistors for Biosensing and where is the field heading to. And before handing the stage to Professor Sabine, I would like to briefly introduce the new journal BME Horizon, and which is an international peer-reviewed open access journal, and it is published quarterly and owned by Science Exploration Press. So we target the initial impact factor more than 5, and it covers different fields in biomedical engineering, including biomaterials and tissue engineering and medical device and synthetic biology, regenerative medicine and nanotechnology in medicine, etc. Yeah, and here this is our editorial board, and our editor-in-chief is Professor Bu Yongma from Shanghai Jiao Tong University, and I currently am serving as the junior executive editors, and we have a broad editorial board from across different countries and different university covering different topics. And then I would like to briefly introduce our speaker, Professor Sabine, and she obtained her PhD degree in 1998 from Queen Mary and Westfield College, and after that, she spent 2 years at the ENS Paris as a postdoc fellow, and after that, she did several postdoctoral positions at different university. We can see across different institutes, and then she became a full professor at the IMP Grenoble, and after that, she was appointed as the full professor at the University of Lille in 2009, and now she has published more than 360 research publications from a lot of papers and different edited 4 books and 20 book chapters in the field of nanotechnology, materials, chemistry, and biosensors. So now let's give welcome to Professor Sabine, and now stage is yours. Thank you. Yeah. So I'm gonna stop sharing, and you may start your sharing, right? Okay.
Excellent. Well, thank you for this nice introduction. Let me get my slides up now. In principle, you should see them.
Yeah,
Yes?
we can see the slides. Yeah.
Excellent. So, well, thank you for hosting me here, and thank you for everybody, wherever your time zone is, to be present and listening to this talk. I thought I'd give a very general outline on the biosensor technology we're doing in our laboratory over the years. So you will see in the following what the interest of graphene-based field effectors will be for biosensing and point-of-care developments. And the reason I want to talk about is, let's simple start. I think everybody knows what we want from a point of care diagnostics. We want that it's handable, portable, transportable, benchtop devices, disposable, and wearable. So you have a variety of different point of care diagnostics which developed over the time. At the very, very beginning, one considered point of care diagnostics more like simple measuring pressure or heartbeats. Now we can consider them more of using bandages or patches on the skin. And you see here on the last image already you can use these sensors eventually also in wearable technology to look at wound healing. I will come a little bit later onto this. So what is different to this point of care diagnostics, if you want, you find in hospitals two point of care diagnostics using biosensor developments. I think the most widely used biosensor for itself is the ELISA platform. So what we use in this ELISA platform, we have a substrate, and on the substrate, we will immobilize something which we call the capture antibodies. Then you will have your analyte. But to read off this interaction, antibody-analyte, you will need a secondary antibody with a readout signal. So that means the secondary antibody is labeled often with an enzyme, and then we look at enzymatic interactions. So what's the big advantage of this sensor platform? Where it's highly sensitive, highly specific. This all goes because we use a capture antibody. But it's rather expensive because we use antibodies. It remains rather time-consuming, several hours, and definitely it's an endpoint technique. It's not a continuous technique. So over the years, if you want different, or I call it the second generation of point-of-care devices, found its place. And one example of this is an electrochemical sensor. So what is the difference to an electrochemical sensor to an ELISA is? Well, again, on an interface, this time on an electrode, we will capture an antibody. Then we will have the binding to the analyte, but we will actually not really require, in an essence, a secondary antibody because we could use redox mediators to read off the signal. So what's the consequence on this? We keep the high specificity and the high sensitivity. We will have fast results because that's the intrinsic characteristics of electrochemical biosensors itself. So it takes about 5 to 10 to 30 minutes before you really have the final signal. It might be cheaper because you only use one antibody and not a sandwich assay, so a second antibody to root it out. It requires redox mediators and it requires potentiostats, which might be large to small for pore sizes. So before I actually show you another generation, sort of an example of an electrochemical point of care device, which comes from our group, which we have just been working on, and I want to use it as a sample. So what I want to show you here, how you use an electrochemical assay for detecting of plant viruses. In this case, grapevine leaf viruses. So what we did, we used the screen printing electrodes. We put antifouling agents onto this because one of the intrinsic problem in biosensors is always antifouling if you measure in very complex matrices, like in blood, but also in this case, actually in plant juice. So we got some nanochemistry based on reduced graphene oxide, BSA, and glutaldehyde. So we get this on the interface. And then we use the remaining NH2 groups, which you have present on this NH2 enriched reduced graphene oxide and BSA, to do our chemistry. And the chemistry we like currently very much is integrating first cysteine, spike catcher modified structures on this maleimide modified interfaces, where then we can more or less interact very easily, like in a biotin-avidin interaction concept, spike catcher ligands. The main difference probably with biotin avidin is the KD is almost the same. But if we would translate it to humans, for sure, we don't have any spike catchers, any biotech in the body present. Like biotin we would have, and even in high concentration. So there could be not an equilibrium between the bioligand attached to the surface and biotin in the blood, which would actually take off the ligand. Fortunately, this is not the case in plant viruses, but that's only like an example. So what we also show here is that the type of chemistry we're using, we can actually also put multiple bioreceptors on the surface. And this is in particular interesting if we go to virus sensing or bacteria sensing, where we always know that there is a mutation in the outside proteins, which we often target for viruses. So in our case, we overcame eventually mutation in this plant virus by adding two bioreceptors on the surface. And to dig a little bit into the bioreceptors, I talked about antibodies, very often used in ELISAs and electrochemical sensors. Here, we took the choice in going away from antibodies and using nanobodies. That means it's the very small binding end of a nanobody. It's around 4 nanometer in size. Using this small synthetic bioreceptors has several advantages. First, they make the assay cheaper in general, but the small size of the bioreceptor also helps actually getting better electrochemical signals because we have less diffusion barriers than we would have with this around 15 nanometer high antibodies. So if we get them to the surface and then we use a plant extract, so you take a plant, you grind them, and then you throw them on the electrode. So in the presence of a virus, we will therefore have attachment to the surface. So what we opted here in this case actually to have a sandwich assay. And the sandwich assay is very easy with a virus because a virus is like a sphere. That means the ligands which are not attached to the interface, they are still present. So we can allow ourselves to attach again two different nanobodies, but this time modified with an enzyme, alkaline phosphatase on the interface. And for the chemists of you, we tried to use a probably novel and more interesting redox label to read off the presence of the alkaline phosphatase. What you do normally in alkaline phosphatase, you need redox labels which are sensitive to the presence of the alkaline phosphatase. But the issue is often that the redox peak of the product is much, much more anodic than to the initial interface. That is a challenge for electrochemical sensors because you will have more fooling interactions and often you probably cannot distinguish between the initial and the product. So what we sort of found, if you use this ferrocene N3-CoP, like you see here, so you synthesize it, in the presence of the alkaline phosphatase, it will split actually into a reaction which is ferrocene NH2. And what you see here in the different pulse photonogram here, so the initial product has a redox couple, a peak at around 0.3 volt, where the product is much, much lower, around 0.1. That means we can rather easily detect the onset of this occurrence of the product in case there is the virus particle present. And to show you that actually this is a viable approach, here are some of our data. So what you see here on the top is an interface which where we put positively charged particles. So there were virus particles in the jewels on the surface. And what you see actually, the initial peak of our mediator decreases and another peak actually occurs. This is definitely not present when you see no virus is present, so you can clearly distinguish from the rise of this E1 peak the presence of the virus. So for this, you can make now calibration curves like you see here. So you get here currently a limit of detection of around 50 picogram per milliliter of these proteins around the viruses with a limit of quantification of 100 picogram per milliliter. So what we did actually, we saw if this detection limit is more or less good enough for real sampling. So what we did, we compared the results of the only commercially virus sensor for this type of plant virus is a DAS ELISA for BioReba, which is based on absorption. And we compared it with our sensor based of currents. So if you correlate actually these different samples, which we have here, The control worked very nicely, so no signal. And then from all these different samples, we had an agreement 100% beside actually one, this T58, which gave a very high signal actually in the DAS analyzer, a very small signal in our electrochemical sensor. So now there could be a variety of speculation while this is happening. It might be happening that the nanobody we are using is not specific interacting towards this, or it might be an issue, a human issue, that when we were grinding the leaves, we sort of lost the particles. We don't know. However, there seems to be a very nice correlation between ELISA and the electrochemical sensor, knowing with the sensor we get signals actually in 2-3 minutes on it. So I hope I have convinced you that this second generation, as I call it, where we use redox labels for readout, is already an advantage compared to ELISA. So now I want to get really more into the core of the talk, which is the next generation of sensors. And these are the field effect transistor, and I will in particular focus on this graphene-based field effect transistors. So what do we have with these systems? We keep high specificity, high sensitivity. We still have very fast reactions time. We still will only need actually one bioreceptor, being it an antibody, being it something else. I will come actually more back to this. But we also don't need any more redox mediators, because we will use the intrinsic current change of the channel of a field effect transistor for a reading. We can use also sustainable materials. gFETs can be integrated nowadays on flexible materials, so they can also be variable. And because of the very small size, we need very low energy consumption, so we need very easy circuits to make this sensor working. So there are different configuration you find in the literature. They can be like top-gated, they can be back-gated, they can be floating-gated. All of them have different advantages. I will not go too much into electronics on it, because I want to focus on the biosensor aspect. In all this configuration, what you can do is you can cover the channel, and this is the sensing element, with graphene. And the reason we believe that a graphene channel for sensing is extremely interesting for sensing is because graphene has very high electrical conductivity. It's very sensitive to charge changes, but it's very, very thin. That means all the charge changes you will see will go through this very thin film. And this actually will give it its very high sensitivity. On the top, you have the chemical inertness of the graphene. You have high mechanical stability. So all this together makes it that you actually have a very sensitive and stable sensor at hand. Now, what are the current research interests or eventually limitations of these type of sensors? One big, the challenge is anti-fouling, and I will come back later to it, actually working with very complex medium like blood. But this is also orientated of the limitation of working in high ionic strength solution. High ionic strength solution like 1 molar PBS, if you want like a model example for the laboratory, but definitely in blood. Blood has a ionic strength almost the same like 1 molar PBS. So why is this limitation? These limitations come to something which is related to the bi-length screening we can do on this type of sensors. So if you do actually look at this lambda delta, you see it is indirectly proportional to the ionic strength. That means as larger the ionic strength, as smaller this LD value. And LD in very high ionic strength solution, like in full blood, would actually mean an LD of around 1 nanometer. So what that means, we can only measure really very sensitively 1 nanometer away from the graphene surface. So this gives now a lot of challenges. And one of the challenges is actually linked to the bioreceptor you have to put on these biosensors. So if we take the example of an antibody, which has a length of around 50 nanometers, I think we all agree we are out of the theoretical sensing limitations of these sensors. Actually, even if we use engineered antibodies like nanobodies or even aptamers, we come definitely closer towards it, to around 3-4 nanometers. We are probably just on the limit. So it will be finally a combination of the right bioreceptor on this interface, but also with the anti-fouling agent, which will give you the right possibility to measure in high ionic strength solution. And here you see an example. You can increase this by length screening field actually by using like polyethylene glycol molecules on the surface. And this is actually validated by some of the experiments I show you here. So if we look at the drain source current of a field effect transistor, if we only have the aptamer, for example, present, we see that around 0.01 ppb, you almost lose your signal. However, if we have a mixture of aptamer and the PEG units, we definitely have a decrease in sensitivity in high ionic strength solution, but we still able to measure at really high PBS concentration. So this will be sort of the guiding rule then the rest of the talk, which type of bioreceptor you can put on the interfaces and how can you put gels or copolymers on the interface to get the biolink screening long. But before this, how we actually physically construct these type of sensors. So currently, what you have to do is you have to transfer CVD graphene on the interfaces. The way we do it, we buy, or for the moment, actually, we even do laser-induced graphene interdigitated electrodes, and you put your graphene on it, and you can use then the drain and source current of these interdigitated electrodes as part of your transistor. So the devices they are all commercially available. You can buy the cell from Microx. If you do this type of approach, that means the U gate will be a simple silver wire electrode. You can buy these source meters, so you come to a point of care if you want, which is a benchtop based device. Now, when you do this graphene of the transfer, it's a wet transfer, so you have to put actually polymer on the graphene that you have it mechanically stable enough, and then you like print it on it. So it's very important that we have a full removal of this PMA on the interface, otherwise we will not have access to graphene to do proper surface chemistry. So you can actually validate this partially with Raman spectroscopy. So the Raman spectroscopy you see here, you definitely have a very nice G band here, but you have a small contribution of the D band, which is towards to this transfer of your graphene on your interface, which is linked to strains. What you probably don't so well is eventually impurities of this PMR surface, which you could see from XPS. But also, if there's only some percentage, you might not know what's part of the oxygen functions on your graphene and which comes from the removal. What you can do for readout is very different. So what I will show you here is mostly actually we do look at the change of the Dirac point to electron rich or to electron whole maturity carriers percentage to see how the sensor works. But before we do any sensor, we need surface chemistry. So we have to put these bioreceptors on the surface. So there are two approaches possible. You have non-covalent approaches with pyrene chemistry, and we have covalent approaches where we mostly use diazonium chemistry. So in the following, I will show you some of the approaches we use in the lab in both approaches, and then you will see the advantages and disadvantages of both. So let's start actually with this covalent functionalization, where we use often diazonium chemistry to covalently link to form carbon-carbon bonds on this graphene by taking off this nitrogen functions. So by doing this chemistry, which is known over years, is we know when we do diazonium chemistry, we can also form polymers on it because it's a radical chemistry. So what one normally uses is a diazonium ligands, which is a bulky group. That means you sort of limit the formation to a monolayer. And this you can easily see here in the cyclovoltammograms here. So the first cyclovoltammetry clearly shows you the kicking off of the N2 group and the first linking step onto the graphene. And then you almost lost all the current. That means actually you really limited to a monolayer modification of this graphene. Now, what's extremely interesting is when you look at the gFET characteristic and the drain source before and after the modification, what you see actually, the initial graphene had a very low hole mobility. However, we sort of recovered this completely with this diazonium chemistry. Now we get the hole and electron mobility, which is around 1,600 centimeters carré volt seconds. So the question is why this is actually happening. So we looked a little bit more into the mechanism, into this diazonium chemistry on the graphene interface, and we used Raman, and in particular Raman image, to get more ideas. So if you actually look at the Raman images, the intensity of the do G over G and the dG band, you see there are not so much differences on the interface. But you can use this equation here, and actually you can calculate not only the defect size, but from the bandwidth of the dG band over the G band, you can actually see that from pristine graphene, if we do our dip modification, we sort of move to the right, what actually means we will have sort of p-doping on the interface. Then we look at the defect size calculation. We estimated around 10 to the 12 centimeter minus 1, which is actually rather low. So we have really a low estimation on defects on the graphene. So what that actually means is that probably what we do is we clean our interface off from eventually some polymer which left on the surface with this diazonium radical chemistry, and therefore get very well characteristics for gFETs. Now you can ask me, what do we do with these diazonium modified interfaces? Well, if we use this TIPS chemistry, you will need to deprotect the TIPS group. And when you do this, you end up with a triple bond. And now every chemist will know triple bonds can now be integrated with diazonium chemistry, and we used here N3 modified ferrocene molecule as a model compound to show that it actually works. So we did click chemistry on this graphene surfaces, and you see the presence of the graphene. You easily see the integration of this ferrocene on the interface. And I hope I convince you with this charge transfer characteristic is that we keep very high charge mobility. That means this process is very well adapted to get very high sensitive gFETs for the sensing approaches. Now I want to show you what eventually will happen if we don't do diazonium chemistry, but if we use non-covalent modification. So what you see here is we tried for a long time to see what will be the best pi-pi stacking interaction chemistry on this type of graphenes. So we compared p-base with the acid form with the p-maldimide form. And what we did, we followed again over time the intensity ratio in the Raman. And what you see here, using simple the p-base, is that when you leave it over a long time, we will change from a monolayer actually to bilayers on it. So what we did then, to make this a little bit more analytical, once we have integrated the p-base on the interface, we again did chemistry in it to get an idea about what's the amount of ferrocene units we can get onto the interface. And here everything gets a little bit interesting now. So if we compare now the p-base, and that's the green part here, first on the AFM, what you see here already after 2 hours, you rather have a rough surface. That means definitely this p-base is integrated. But then if you look at the chemistry where we put the ferrocene on it, that's quite surprising. We sort of get ferrocene units into it, but over time, actually, the concentration of the surface gets much lower. With too long at times, like 12 hours, we actually see nothing anymore. If we then compare this to the acid format, you also see that after 2 hours, you easily have already some of the structures present. With 24 hours, seeing actually already holes and the more 3D structure. Here again, when we put ferrocene on it, we easily can, even after 24 hours, still see ferrocene coupling. So what that means actually, the P base, which an ester, seems to be not very stable on the interface, that after 4-6 hours, it gets hydrolyzed, and then it will not allow further bioreceptors being integrated on the surface. So it might be not, even so widely used, the best approach to modify graphene-based. It can be only competitive to other chemistries if you really control the time to a very short time. Then if you compare this to the acid, yes, here we keep the acid over time, so you cannot hydrolyze it. But what you see from the AFM, that over time, instead of covering the whole surface, you will probably more get a 3D structure on it because the pyrene units stack over it. But there will some holes be left, and these holes actually would mean you will have a direct access to the graphene interface, which is not ideal when you want to work with media like blood or plant juice, because you will have a lot of problems with non-specific absorption. That's the reason we believe, and in all our work from this time onwards, we use maldemite modified pyrene ligands on the surface. Because what we see here is we can easily integrate the bioreceptors in a concentration-dependent manner, in time-dependent manner, over time on the interface without losing actually the sensing possibilities and having a very nice and thin film. So that's enough now on the chemistry for itself. So I want to go now to some examples, what you can do with these interfaces. And one of our first example was linked to something which we call diabetic foot ulcers. So diabetic foot ulcers is sort of a side effect of diabetes. And why are we so much interested in this diabetic foot ulcers is because if you see from this card is that there's a prediction in the World Health Organization that diabetic patients will drastically increase in the next years. So what you actually see here is depending on the countries, for example, in the Middle East and North Africa, there's almost 100% of increase of diabetics overweight. But even in Southeast Asia, you have up to 74% increase on it. So how is this increase actually related to us? It's mostly linked to the bad lifestyle we are having. We are eating too much fat. Probably also too much fries in Europe, because even in Europe where you see it's a 50% increase. So the area in Lille where I'm coming from, it has a really high increase of diabetes over the years. So what that actually means is that the health expectation and even life expectation of our children is much more decreased to us. And it's not only a problem of decrease of life expectation. Diabetes has a variety of side effects. You can have side effects on the eyes, increased strokes, nerve diseases. Here I don't want to go into, but one of the biggest side effects actually is this diabetic foot ulcers. That means it's unhealed wounds which develop on the foot and cannot heal over years. So the idea is actually to find bandages you can put around the wound, which give you an indication actually of the healing state of the wound. So we thought graphene field effect transistors might be just well adapted on it. And I quickly guide you through how this chemistry is actually working on this. So what we opted for is an aptamer-based sensor. So this aptamer-based sensor is combining some of the ideas I explained you beforehand. We don't want to use aptamers, which are rather large. We want to use two small structures where we can measure in complex media. In this case, it would be wound fluidics. So if you look at the literature, there are some aptamers related for wound healing, and one of the aptamers which are indicated is an aptamer for this biomarker called MMP9. So MMP9 is actually overexpressed when you have inflammations and bad wound healing presence. So an increased level of MPP9 would therefore be an indirect indication that something is not working well in your wound, and therefore you will have to have some changes in the treatment. Now, for probably people who are also not so familiar with aptamers sequences, they form functional aptamers because they go into a three-dimensional structure, but then when the biomarker actually binds to it, we have conformal changes and therefore conformal recognition. And because it's a charged species, you will have charge effects taking place in the sensing interface of the graphene, and this we are going to measure. So how was this MMP9 aptamer linked to the surface? Well, it was linked to the pyrene maldemite chemistry, which I just showed you beforehand. And here is the sequence of this aptamer which we used. So let's go back to the sensor itself. So to directly going towards this, we used simulated wound fluidics, which is already a very complex structure of high ionic strength. So what you see here, if we follow the change of the current going through the drain source over the concentration of MPP9, you see up to 400 picomolar, we will not have a change of the signal, but then we clearly have the Fiedefeld transistor responding. So what that means is in the simulated wound fluidics, we will have something around limit of detection of around 500 picomolar with a linear range definitely larger than 1 nanomolar. You see immediately actually the complexity working in complex medium, because if we do exactly the same sensor in simple MOPS with a very low ionic strength, we will actually get a detection limit which is 4 or 5 times lower than this one. So the question is, would we be competitive with this type of sensor characteristic compared to an ELISA? So we had a little clinical study. So we took wound fluidics from foot ulcers from patients, men, female, different ages, different diabetics. And we looked at the overall protein content, but also we performed ELISA on it. So what you will see clearly is, for the moment, ELISA assays are still more sensitive because they can pick up very low levels of MPP9 actually in these wound fluidics. But then the limit, which makes it actually interesting for measure, where you really see that something happens in the wound, is around 200, 300 picomolar. So this is actually where the gFET actually seems to be competitive with the ELISA. We almost get the same concentration method between our gFET and the ELISA, with the advantage is actually that now we have a reading in 1-2 minutes. You can integrate these bandages onto the wound and get a direct online reading time. But also an advantage of this approach is, and I know it gets a little bit out of sensing, but I will give you some idea of the importance of this type of devices is, you can not only measure it, but you could probably consider integrating these sensors into matrices, jails, which measure biomarkers where you do the sensing, but at the same time also will help you healing the wound, because sensing that there is an infection present is one thing, but then treating the infection is another one. So in a work in a European project called G-Skin, we tried to make this diagnostic approach and focused also on the development of gels like this hydrogel you see here. Actually, it's not only hydrogel, it's based off of the gelation in the presence of sodium bicarbonate. And because you have sodium bicarbonate at a certain pH, you will generate CO2. And these CO2 bubbles can actually get impregnated in the gel, so you actually get a foam. And in this foam now you have a very high surface area. So what we did here, we integrate proteins, and proteins actually blood-derived extracellular particles, which have shown to be really adapted for wound healing. So we can load these gels, we can release them, and then we actually can look at wound healing effects. So I don't want to go too much into details here, but these were the rats. They had diabetic wound ulcers on it, and we showed actually by applying these bandages on them that we get really high wound healing with our gels having integrating these platelet-derived particles. So I think it's clear for you how this diagnostic approach will work. You will have the sensor which tells you there's an inflammation going on, and then we will trigger the release of these platelet-derived particles to the wound to have healing. The other example I will quickly want to go through in the last minutes is all our work we did on the sensing of CTNI on this type of structures. Cardiovascular diseases are increasing every year and almost become first cause of mortality. When you have a cardiac problem, you will get to urgency, and what happens in urgency? You will probably most likely get immediately this electrocardiograms. But the problem with electrocardiograms is very often nothing actually is observed, so the patient is sent home, and then actually when they're at home, they will get a stroke. So that's one of the reasons other assays are requiring it. You will definitely eventually not get immediately a CT scan or electrocardiogram scan, but what you could get immediately would be a blood test. So again, it's an ELISA blood test, which measures very sensitively CTNI in your blood. Now, as I explained to you beforehand, the problem with ELISAs, and especially in a clinical turnaround, is it's also expensive, but it's very time consuming. So it takes a lot of hours before you gave your blood to the time actually you get the result. So 2 words about the biomarkers. For example, forcingly, you can choose several biomarkers. We decided to go on troponin T and E. And the reason is the following, as you see here. When you have a cardiac problem, most people, even before they would go to hospital, it will take them some time. And this is a biomarker which takes about 3 to 6 hours to onset of the pain where you actually have the increase. So you might be in the best position that you have the maximal level of this biomarker in the blood currently when the patient will be received in the clinic. So that's the reason we focused on this troponin. So the next thing what you have to take into consideration are detection limits, because this will be important when we design our sensor. So normal people have levels below 15 picogram per milliliter of this CTNI. Small cardiac damage are between 15 and 500. And then if you really have high levels, you're already at very, very high risk. So what we want to do with our sensors nowadays, we really will want to be able to distinguish between the 3 levels, between people who are not ill, between people who have probably an onset of damage, and between people who are very high at risk already to have CTNI in the blood. So we constructed our gFET sensor. Now you understand the chemistry. So the chemistry we used here was this click chemistry, and then we used the CTN-aptamide on it to have a small ligand, but we also added PEG units that we can allow to work in blood in high ionic strengths. So here you see all the data on these field effect transistors. So they work very nicely. What you get here is a limit of detection of around 3 to 4 picogram per milliliter. So we are really in the detection range that we can distinguish between healthy and onset people with a linear range to around 400 picograms. So we just actually make it to tell you if you're really a high-risk person. So this is what we did. We used the samples in a clinical study, and to get the ionic strength a little bit down, what we did, we put blood through some desalination columns. And doing so, we could finally actually distinguish between low, medium, and high levels of troponin patients. And you see here all the different numbers. So we also compared them with ELISA. So we clearly could, like ELISA did, classify the patients into the three groups. The red group being the very high group, then the middle group, and then the ones we couldn't distinguish at all. Now, let's go to the next step, and I quickly want to show you things we were working on. As I said, in this type of work, we used desalination columns, which is not ideally adapted still for point of care. Ideal for a point of care for CTNI, you would use a fingerprint, you put your blood and put it directly on your sensor. So our last work actually was linked to how can we make an interface which works under these high ionic strengths being not fooling and stable over time. So we constructed a sensor based on this copolymer. So if you see this copolymer here, it has two different surface function here. It actually has been shown in the literature that it has antibacterial properties, but for us, we will show actually that it has anti-fouling properties. And what's important as well, it has possibilities of anchoring bioreceptors on it. So the first question is, would this type of matrices go onto our gFET channels? So what we did the synthesis of a pyrene-based copolymer that it makes it easy, simple for surface functionalization. And this is what you see here again on this Raman images. So you see here that the intensity of the G-band ratio changes significantly when the hydrogel is present. So that's one of the first indications that we have a layer on it. You can go to our paper, then we have also other techniques like SBR and QCM, which show that the gel is really on the interface. But let's go to the things which are important for us. It's the base screening and the anti-fouling properties. So what you see here is actually we keep extremely nicely the gFET characteristics in different ionic strengths. But when you leave this type of sensor up to 120 minutes in full unprocessed blood, the signal is not changing. So we hope to have overcome both problems, the debailing screening and the anti-fouling strategies. So we therefore went into sensing. We had this shell on the surface. So we did again some chemistry. So we added some triple bonds on this COH over protonation first, and then EDC and HAs linking of this type of ligand. We clicked our aptamer on it, and we locked into interaction with our biomarker. And as you can see now from this curve here, when we do now the measurement simple in full blood from a blood droplet, we can distinguish clearly between low, medium, and high CTN levels, and we can even get calibration curves. And if you look at these calibration curves, now we have detection limits of around 1 picogram per milliliter, so clearly adapted for distinguishing healthy from unhealthy people, with an increased even linear range up to 1,000 picogram per milliliter. So if you do, I didn't show it here, we scan several blood samples, so we are now in a position in 2-3 minutes using these devices, if you want, taking a sample from a fingerprint like you would do for a glucose sensor from a patient and detect your CTNI in the full blood. So I want to finish now with some future perspective that you see what one can still do in this field. One thing which I think is very interesting in the field, probably considering different bioanalytes. I explained you all the problems with high ionic strength medium. So why simple not changing the medium where we want to detect our biomarker? And one useful sample might be X-hold press condensate. So X-hold press condensate, is actually press, which when it travels through cooling units get condensed. And in this type of press, you will not only have small volatile molecules present, but also protein, virus particles. That makes the approach definitely interesting. And this is what we could sort of show more recently, again, on cardiovascular patients. If we collect the press in the form of a press condensate or masks, which you can cool down, and then we dose the pro-NT-proBNP level in these patients, we again could, if you compare the plasma between the EBC level, see a certain correlation between the EBC and the plasma. And what's interesting, actually, stage A people, that's actually people who have almost no cardiac problem, we see nothing in the EBC, but at higher stages, actually, we get more and more pro-PMP biomarkers in the press. That means this could be an interesting alternative of thinking for some new biosensors. The other possibility is changing the bioreceptor. I think I show you already examples of nanobodies. I show you examples of DNA aptamers. But if you look at the DNA aptamer, it stays its charged species. So you could always say, well, there might be an interference of the charge of the aptamer itself with the readout signal. So probably ideally you would look more for a non-charged bioreceptor. And there are alternatives to these DNA aptamers. There is DNA aptamers, which have no charge. And this is some of the work we are just currently finishing with our colleagues in Italy. And it's the group of Roberto Carondini. And what we could show actually here by using now not gFETs, but the surface plasmon resonance studies, that not only that we can understand better what the sequence would be needed for this type of PNE aptamers, because the difference to PN aptamers to DN aptamers is that you cannot use the Salix process actually to develop this. So what we did here, actually, we used the same sequence that we had for a DNA aptamer and converted it into a PNA aptamer. And in this side of work, we should actually can show if we make the aptamer smaller and smaller, we still can get very nice affinities up to an aptamer of 15. So if they get too small, then we definitely have no binding on it. But we can also show, and this is what we will show in this paper, that these PN aptamers are extremely stable to DNases, which are very often present in blood samples and lead to a degradation of these aptamers over a long time, while in the PNA sensor, you have no degradation. The last thing which I want to show you is, so we had different ligands, different bioanalysis, but we can also use these type of sensors in a completely different manner. And one of our projects we are going on is trying to use these sensors for managing, not sensing, but actually surveillance. And one of the idea was actually how we do manage virus surveillance. And I simply take the example of Europe. So how we do violence surveillance, actually, we do analysis of wastewater samples as well as of pooled surveillance strategies of patients with a weekly up to monthly swab test. So we need up to 300,000 people, and we then do this PCR analysis. And that sort of gives you then an idea if some viruses are emerging. But you understand immediately it's extremely resource intensive. It needs the public. So the idea is if we, with our sensors, we can do different alternatives. So PSCR is definitely not useful for a general surveillance system because it's not an online system as well. So before I show you a little bit our results, so how could we do it on a sensing platform? And then this is sort of a work which came to our attention is there were cells, they were infected with a virus, but then they were stained with an anti-double-stranded RNA antibody. So you see clearly that there is a clean part on the infecting cells and there is no clean part on the non-infected cells. So what that means, probably viral double-stranded RNA is a universal biomarker for virus presence, generated either in the cells or during the viral general replication. So what that would mean is we will have to think about the double-stranded RNA virus surveillance strategies, but currently actually there is none of this too much available. So for a double-stranded RNA strategy, we again need a bioreceptor. And for this, we also looked a little bit in the literature, and there's quite some out there. So how we can easily detect these double-stranded RNA beside DNA. There is a protein, this B2 protein from froghouse virus, which is a homomer in solution, but has a really strong affinity to double-stranded RNA. So you see actually in this nucleic acid mobility shifts assay that it only occurs with double-stranded RNA and neither with single-stranded RNA, but also neither with single-stranded and double-stranded DNA. So this type of the work inspired us to make the first double-stranded RNA sensor. So you see here the concept again in case we used again the chemistry on it. This time we used nickel 2 plus B2 chemistry to link the receptor on the surface. And what you can see here, we can titrate nicely double-stranded DNA with a limit of detection of around 0.32 nanomolar. And we are definitely specific now to double-stranded RNA, and neither single-stranded RNA nor single-stranded DNA are present. So we probably are now finally in a position of having a different proposition on it. And this will be sort of the next experiments we will continue doing in this type of experiments, using mosquito samples, extracting double-stranded RNA on it, and see how far we are going. So I think my time is out with this. I definitely need to thank everybody involved in this project, because there are projects running over several years. And thank you very much for your attention.